Hello, everybody. My name is Jun Wang. I'm an assistant professor at the Department of Medical and Molecular Genetics, while directing collaborative call for cancer bioinformatics shared by IUSM Comprehensive Cancer Center and the Purdue University Center for Cancer Research. Today, I'm going through the workflow of RNA-seq analysis in the call. And here are the steps of RNA-seq analysis we used in the call. And you can see different colors here. For example, the original one represents the original data processing until and to get the high quality mapping rates, followed by the gene expression summarization. And we also need to perform quality control on biological samples in terms of gene expression. And the last part and also the core part of RNA-seq analysis is differential expression analysis. And I will go through step by step with you in next few slides. First is the trimming step, and we use a trim Garo to trim adapter sequence and low quality reads, and followed by the sequence alignment to reference genome. And the software STAR is the default software tool we used in the call. Why STAR? STAR analysis has been proven to be high accuracy with much faster mapping rates. Of course, it also takes intensive memories. The main idea of the STAR is they try to separate the race for different parts. I mean, for every race, the STAR will search for longest sequence exactly matching one or more genomic location or reference genome. Those parts will call the maximal mappable prefix, MMP. So for each part, and we have MMP, we may call it a seed. For this one, if we first found seed 1, then we are going to search only unmapped portion of this and try to find where they are. And if we find this one, and we call this seed 2. Of course, in many cases, due to either mismatch or in there, probably for some part, it's hard to find the exact mapping. Then we need to go back to check the previous MMP to extend the level, some level, try to find the best mapping locations. In some cases, if the quality sequence is not good or there is a adapter here, we may imagine the STAR cannot find the mapping location. So the STAR will use a software to clip this part of sequence. If you are interested in the details about the STAR alignment, you may go check this website. After mapping, and we know some rays are perfect mapping to map to the gene reference genome, but some rays with a kind of level mismatch or the multiple positions, something. So we need to filter out rays with a low mapping quality. And after mapping, normally, and the BAM file will be generated binary alignment map. BAM file cannot read bias. However, many smart people are working in this field. We are happy they are here. So some people develop one tool, same tools, which are frequently used by bioinformaticians today. We can convert the BAM file to SAM file, sequence alignment map file, which can be read by us. This is the head of the SAM file, and under the head there are millions of seeds there with the important information after mapping. After mapping from this one, we know this sequence coming from chromosome 20 at some locations, followed by a number here. This number is a mapping quality. It's the probability of mapping position is wrong in the format -10 times log 10. In other words, for example, if this is a 20, so which means the mapping position is wrong, the probability is a 0.01. We use an software, NGS utils, to remove those risks with a low mapping quality. Normally, people have a very reasonable cutoff, like a MapQ less than 10, and also that's the default set in our core 2. After we get a high quality mapped risk, we want to know where they come from exactly, because this is a messenger RNA-seq. So we want to know which gene the risk come from and how many risks on each gene. So we need to summarize or evaluate the gene expression given number of risks mapped on the gene. And we use the feature counts to get the gene expression profile. Right now, many experiments, let's say over 90% experiments, they have multiple samples under different conditions. So from that one, we need to perform another level quality control on biological samples in terms of gene expression. We want to know whether different group of samples can be distinguished successfully, or we want to see how big difference between different group of samples. So we need to perform quality control to make sure they make sense in terms of biological meaning. And the last part, also the central part of messenger RNA-seq analysis, is a differential analysis. And we use the XR to do differential analysis, try to identify genes which are differentially expressed in different conditions, under different conditions. However, most times before the quality control, we have an additional step we call the normalization. As I mentioned, we have multiple samples coming from different conditions. If we want to fairly compare them, we need to do some level of normalization. We need to put all the gene expression from different samples, from different conditions, they are parallel. And you may ask me why. In the first step of gene expression evaluation, we already normalized the race gene expression by total counts of race, like we use the RPKM or CPM, count per million. Both are normalized by total counts of race. However, please remember, total counts of race doesn't represent the true measurement of total RNA outputs sometimes. Particularly when few genes, they are very high expressed in one individual sample. So there are several normalization methods. Actually, there are many normalization methods people use. Several main normalization methods are quantile normalization. Quantile normalization is to force distribution of gene expression to be identical for all samples. Sounds very aggressive, right? And that's right. And especially some biological experiment doesn't think quantile normalization makes sense. And another one is kind of very loose or very flexible normalization we call the log geometric mean. In other words, people just set the mean value of all gene expression for all samples to be the same. And there is another normalization with the hypothesis. Majority of genes between the samples, they are not differentially expressed. So in that way, people try to minimize the log fold change between the samples for most genes. Then they can get a scaling factor for library size of samples due normalization. In the C3B core, use the trimmed mean of M value, TMM normalization as a default one. This method comes from the assumption that most genes are not differentially expressed. The method was developed in 2010, published in Genome Biology. Here, the picture, I copy-paste the picture from their publication. In their study, they tested RNA-seq for two tissues, kidney and liver. And they found for the same tissue, the fold change or difference between the samples are centered around 0, which makes sense. So that means majority of replicates, biological replicates, they don't have significant change. However, when they compare liver versus kidney, and you may see the distribution shifted to one side, in this case to the negative side. 0 is here, including the green line representing housekeeping genes, which means majority of genes in the liver, tested in the liver, has a lower expression than in kidney. This is a MS plot, which shows X as the average of gene expression across two tissues. Y is the difference for the change of gene expression in two tissues, liver versus kidney, and each dot is one gene. It's a more detailed representation to this one, but basically they are same. You can see majority of genes shift down, which means they are lower expressed in liver compared to kidney, including housekeeping genes. That's because some genes here, they have a very high expression level in the liver based on simple normalization by total counts of rays. Then, because they occupy the majority of rays coming from this part, so other genes, they were under-evaluated in terms of gene expression. Then we should think about this factor and do some level normalization and try to identify or calculate the scaling factor for the library size of all samples and do renormalization. HR is very commonly used in differential analysis, which is also default method in our core. HR is an empirical analysis of digital gene expression data in R. HR implements many statistical methods for different comparisons. For example, they have a negative binomial dispersion model and the generalized linear models and the empirical based node models with multiple flexible options. HR can be applied to differential analysis at a different level: gene level, exon level, or transcript level. In fact, read counts can be summarized by any genomic features. And also, people are developing HR right now. And so right now, the HR analysis at the exon level are easily extended to detect the differential splicing or isoform-specific differential analysis. And if you check online, and you may find that there are over 100 pages for HR manual, but I cannot describe all details in this lecture. In the next one, and I will show you some level demo of RNA-seq analysis in the IU supercomputer system. Thank you very much.